So, as Jerry said, I come from a very different background to most of you. And so this is a first for me, even in terms of scale, walking this morning into a presentation where you have 900 people there. And many of you know more than I do about pigs. So today I'm going to be on safe ground and we'll talk about molecular biology. And hopefully I'll try and introduce you to what I think a molecular biologist should be thinking when it comes to swine research. I'm also going to try and give you an idea of the sort of thought process one needs to go through when trying to embrace molecular biology. And when I talk about molecular biology, I'm talking about sequencing. So I think everybody knows how sequencing has changed. And we went from an environment where we had, you could only sequence on a daily basis, 100 base pairs sort of sequence. But now you can generate gigabases of sequence, and you can do this within a few hours. And so the world has changed, and it's changed literally in the last, especially for the last 10 years, where we've had an explosion of sequencing techniques and tools. And this is what is known as next generation sequencing. So I've come from a lab where I was brought up using lab sequencing, slabs, and moved into the automated side with ABI and the Sanger sequencing. But I've also now, as I say, starting to embrace this new technologies of next generation sequencing. And, the title of my talk is about the present and the future, but to be honest, the future is here. These technologies are here, and it's going to change the way we diagnose and understand disease in most systems. The other important point to make is that, as you can see, sequencing has been around for quite a long time, since invention in the 1970s. It's never really taken the lead in terms of sequencing, has taken the lead in diagnostics because of the time it takes, right? It just takes too long to try and get a sample and get it sequenced and analyzed, and then for you to identify what the pathogen is or what's present. But again, as I say, the future is here because we can start generating data within a few hours and understanding what's present in the sample within a few hours. I can also say that the technology is moving at such a pace where you can actually take these samples into the field, collect samples in the field and take the sequencing tools into the field, and you can literally sequence in the field. So, as I say, the future is here. The question is, can we embrace it and can we use these technologies? And I've borrowed this slide from Jerry, and this is something that the VDL here in Minnesota would think about when it comes to pathogen discovery, for example. And if you work with them and you supply samples, these are the sort of things they will be proposing in terms of what you would do, right? Whether it comes from classic virology or bacteriology or serology. Sometimes you might want to have a look at a sample under a microscope and using electron microscopy. But as I say, more and more there's this development in terms of the molecular development as well as molecular diagnostics. And I say that the world is changing because the sequencing platforms are there. So to get a feeling of where I'm coming from and what I'm thinking, I've pulled up this slide because, as Jerry says, I'm new to the university, I'm new to the faculty, and I've come from the UK. And moving here, you get a little stressed, I guess. And my wife pulled this, got this email from a friend, and she said, have a look at it. It's a YouTube video. It's only a 1-minute video. If you look at it, I think it's worth looking at. And it's titled, Then Why Worry? And this is a guru named Hassan Maghboul, and he comes up with this theory as to how one should live one's life. Okay? And it's based on this slide. So, do you have a problem? Okay? And if your answer to that question is yes, then you ask yourself a second question, can you do something about it? And if your answer to that question is yes, then why worry? Because you can do something about it. Okay? Or if your answer to that question is no, well, you can't do anything about it, so why worry? I think it's a great philosophy, right? And of course, if you don't have a problem, then why worry? But actually, if you check and convert this to my world is, if your animal is sick or if you have a problem, sequence. You don't have to have a theory as to what's going on and what you think is causing it. Just sequence it. And the answer to all these questions is really actually sequencing it. But the reason why I'm showing this is actually what I would like. I think Stephanie mentioned this in the first talk, and I've heard other people mention this in talks here today, is that more often than not, we don't have a baseline. We don't know what is out there. The majority of our diagnostics is reliant on disease and well-characterized disease and what we think we know about disease. And we go about understanding and designing experiments based on what we know. And more often than not, especially when it comes to sequencing data, we don't know what the background is like. We don't know what is out there. And usually the answer to that question relies on when you think you don't have a problem. So actually, this is really key. We should be sequencing now when we don't think we don't have a problem. If you don't have a reference, you can't compare when new emerging viruses, new emerging pathogens come into play. You have nothing to compare to. So I think somewhere down the line, we really need to get down to this point of sequencing everything. So when do you want to make a decision of sequencing? Okay, so as I say, I would say all the time. But in terms of your practical reasoning behind why you would sequence something is generally you see a disease in your farm, or a farmer sends you a sample as a vet to say, well, something's going on. And then you have to make a decision as to what you would sample. And there's a list of various samples that you could take, and that's generally based on your understanding as a vet or as a practitioner what it is you need to sample and what you think is appropriate for sampling. But I also do think that you need to think outside the box and actually take samples that you think might not be associated with the disease. And something I do, coming from being a viral ecologist, I do think one needs to sample your environment. And I think this is something which is really neglected in terms of what is in the environment at the time. Okay. And then, of course, when it comes to your decision, and once you've decided to sample, to send something for sequencing, most people would use a protocol for sequencing which allows you to capture most things. But actually one quickly realizes how this can change. So you think as a decision tree, this is pretty simple. Get a sample, send it to your viral diagnostic laboratory, and you sequence. But in reality, people make various other decisions along the line. And I think the majority of people would say, well, it would be nice to know what it is really that's causing this problem. So let's go and isolate the pathogen first. And if you can do that, and if it's something which is recognizable, generally because you think you know what the disease, the causative agent is, you go and sequence it. Now clearly, once you've made that decision, that's very different to just sending a sample off for sequencing. If you made a decision that you want to isolate something, what you only can do from this decision line is that you can only get information on a genome of a pathogen you think is causing a problem. You immediately make an assumption that anything else in that sample has no role to play in disease. So you've already from the start, your very first decision is actually to eliminate everything else. And sometimes I think you need to make a decision, well, actually it's maybe worth sequencing everything and, of course, sequencing the pathogen you think is causing the disease. But you also need to make a decision as what you're going to want to sequence and what you're looking at. Again, the answer is pretty simple because you would say, I know that the pathogen that's causing a specific disease is either an RNA pathogen or a DNA pathogen. And so you make a decision again, is it DNA or RNA? But sometimes it's actually worth you doing both. Because even if it's a DNA pathogen, you don't know, and you get a presence from the sequence data saying, yes, I can see 20, 30 different virus genomes and 20, 30 different bacterial genomes. It doesn't necessarily tell you if any of them are actually active or doing an infection. You can get something on the load, but you don't really get information of whether they're actually doing anything. Now, if you do sequencing and RNA at the same time, you actually get a feeling for whether something is actually functionally doing something. It's actually replicating. Okay, so there's an advantage of doing both. But again, I suspect most people would end up going down this path of isolating genomics and doing it and requesting a DNA or RNA if it's an RNA organ pathogen. If you do that, then you have to make another set of decisions. Right? If you decide, well, actually, I want to see what's going on in the whole community, you can barcode using PCR, and then you get yourself what you call a microbiome now. We've been doing microbiome work for a long time, actually. This is labeled under a different name. It's called microbial diversity. But now everybody's doing microbiome work. But you can do this, as I say, direct through PCR, through barcoding. Or you can do the same thing through total DNA. You can do an amplification step. You can create a library. And you can also do sequencing and do a metagenomic sequence library. Again, that gives you total diversity. Or if you go along the pathogen route and you choose the barcoding route, you end up identifying the pathogen you're most interested in. And of course, on the other side, you do a similar sort of thing when it comes to the RNA. But with RNA, you have to convert the RNA to DNA first. And from the DNA, you then go and do your viral discovery or your bacterial discovery. It's only now, and actually the last month, you can actually sequence RNA directly. Okay, so this avenue is something which didn't exist about a month ago. So there's an option now where you don't have to introduce this additional step. And for certain pathogens, this is really ideal, especially viral pathogens. So again, the decision tree as to whether to sequence or not can become quite complicated. It can also mean that your decisions you make early on will determine what sort of questions you're going to be answering. And as I say, the other point I'd like to make is that if you do use the RNA route, this sort of method in terms of transcriptomics or RNA-seq is highly valuable when you want to know whether your DNA pathogen is actually doing something. And again, I can't iterate that more by saying, sometimes you should be thinking about diverse routes of sequencing when it comes to the sequencing tree. But of course, it doesn't end there, because you also have, as I outlined in that first slide, you have so many decisions to make in terms of how you go about sequencing. The leader in sequencing now, especially next generation sequencing, is Illumina. They have so many different platforms, and that's based on very much the sort of depth and the timing in terms of what you want back from your sequencer. But there are other competitors out in the market, is PacBio and Minai, and this is the new technology which allows you to sequence RNA directly. But more often than enough, it's appropriate just to stick to Sanger sequencing. It's still a good way of doing things, just because of sequence accuracy. But you see the complication of when I say go and sequence everything, I mean to sequence something, there's a lot of decisions you need to make when sequencing something, as I say. But the biggest problem, I guess, when it comes to sequence data is the bioinformatics side. So you can generate all this data, and that's where most diagnostic labs struggle and trying to get the data to you, because the time it takes to analyze the amount of raw sequence data you get to get any sort of meaningful data to you. That said, the bioinformatics field is improving all the time. And again, as I say, the future is with us. You can now buy off-the-shelf sort of computational software where you can transfer and transform your data into something meaningful within hours rather than within months. And a lot of that is based on whether your question. You can go through a bioinformatics pipeline if you really know what you're looking for when you do things like we call reference assembly. You can do a lot of this work very quickly, as I say, if you target it, and you can get your result within hours. But if you do the more fundamental mechanistic approach, it's still a matter of weeks and months when you do things like de novo assembly. And more often than not, based on most things with sequencing, your sequence data is only generally as good as what you can compare your sequence to or with. Right? And more often than not, you have to generate that comparative data sooner rather than later. So I'm going to give you an example. I've come from this marine laboratory where I've been in the UK for 17, 18 years. And my background, say for 17, 18 years, has been about looking at marine systems. And the advantage from my side of things in terms of this training is that we often, especially in the marine environment, we can't see the organisms we work with. And so we use sequencing as a tool to, A, identify them, but B, we design our experiments using all that sequencing pipeline options to try and discriminate who's doing what and when. But funnily enough, an example, and so I've sequenced a variety of pathogens. We've discovered new pathogens doing it this way, things that people thought never existed. We've sequenced even the genomes of the organisms that these viruses infect. Again, things that people didn't know existed 40 years ago. We've sequenced viruses that sit in genomes, that hide in genomes, that are latent, that people didn't think existed before. We sequenced those and we identified those. We can then say, well, actually, the sequencing technology is so new now, we can sequence everything. So now I didn't just work as a virologist, I worked as a general pathologist. I was able to sequence viruses, bacteria, protists. And more recently, we were able to look at areas that we've never been able to sequence before. So we've published sequences generated from water collected in the Antarctic. So again, it's something we don't actually have to see the organisms. We can detect what they're doing, where they are, without having to see them. And then you can design various probes and various primers and look for where these things really are. And here's one example where we've just recently published 2 months ago, viruses in kelp, in our big seaweeds, that previously thought didn't exist. Okay, so sequencing allows you to find things that you think people say there's no problem, we don't see disease, but actually they're loaded with viruses, and the question is, what are they doing? But the example I'm going to talk to you today, and give an example of what we're talking about sequencing, is work I've been doing on honeybees. And honeybees is a very, well, we know the significance of honeybees, but it's quite an interesting example because honeybees is a scenario where you have an infection, okay, and the outcome is a colony dies within honeybees, right? And more often than not, it's asymptomatic. You don't know what's going on. There is no reason as to believe why a honeybee would go and just crash, okay? And that then means that you can start thinking bioinformatically and using diagnostics as a way of overcoming this problem. And so it comes to the question of looking at controls. But the problem with controls is that because it's asymptomatic, you don't know what's going on. All you do know is you can compare the end product, right? Whether a colony is either disease-free, it looks healthy, or in this case, you know it's healthy. But the problem is that these two scenarios look the same, but the difference is one has an infection and the other one doesn't. And so it's very difficult to just by diagnosis, identify what a pathogen is really doing in a system like this. But bioinformatically, you can. And as I said, the example I'm going to give you is the example of the honeybee. And we know in the US, there's been a 60% decline in honeybees since the 1940s. We know in the press, there's been arguing on the pesticides, various environmental factors. But there's a large proportion of this disease has been associated with pests and pathogens. One specific pest is this varroa mite, which feeds on honeybees. And we know that this varroa mite spread from the east and jumped to the various continents, especially in the US, more recently in the 1970s, 1980s. What this mite has done is to transfer this one virus. I'm not going to explain to you why this virus is important, but please trust me when I say that this is the virus causing majority of deaths of honeybees. And the phenomenon, and so in fact what we do is, what we sequence healthy bees. We do not sequence dying bees because that's almost the end product. We sequence honeybees that look pretty healthy and they fly. And as I said, this is the scenario where they were healthy and a month later the colony collapses. And generally the colony collapses mainly after overwintering periods. So this is a survey that's been going on within beekeeping community for the last 12 years. And there's essentially it's consistent 35 to 35% of colonies are collapsing over wintering periods. And total colony collapses is averaging out between 40 and 45%. And the majority of beekeepers can recover from scenarios where you can get collapse of, say, 15, 20%, but when you get to numbers of 30 and 40%, it's really difficult. In fact, many beekeepers are going out of business now because they can't cope with the collapse. Now, to understand this scenario is that of viruses and virus-infected viruses, is that actually you should not think about viruses as an individual strain. And in fact, the definition of virus species is very complicated. but essentially multiple genomes can make a single virus species. Okay, that's important. And it's important that also that certain virus species will occupy a certain environment, a certain niche. And that's also important. Again, there's a lot to understand with the system, but also one needs to understand, especially with RNA virus, is there's this additional complexity known as a quasi-species. And that means a species can have a lot of various diverse sequences in them, but together the different genomes act together to cause disease. It's not one strain that causes disease. It's this complex of a quasi-species that causes disease. I mentioned this, I want to explain the quasi-species. I mentioned this is because when I started working on honeybees and the honeybee viruses, it was assumed there was one virus which encompasses this one virus strain that encompasses this thing called deformed wing virus. In fact, due to genomics and sequencing, we're able to split this virus into 3 groups, A, B, and C. Okay? And when you do sequencing, when you do sort of more high-level sequencing of the genome level, this A, B, and C fits into distinct groups and clusters. They all potentially could cause disease, but bioinformatically we know they cluster in a distinct group. So the question we had is, do these specific gene clusters or quasi-species, could they cause problems in the honeybee? And this is one of the biggest papers we produced, I say that science paper, where we went to Hawaii, and Hawaii had a recent introduction of Varroa. And when Varroa entered, the virus levels went from background 10 to the 4 to 10 to the 10 viruses per honeybee. Okay? And that was when Varroa entered Oahu, the island of Oahu. And the other islands where we never had Varroa, the levels were pretty low. And this happened on the second time when Varroa then jumped from Oahu to Big Island. Big Island went from background to increased levels. So if we had to, so this is just virus load. Now if we had to stop just there and say, well actually CT, or some people say, well let's look at CT. CT will tell you a lot whether something's infected or not. Well, the CT would say this is really low, so it's low numbers, it must cause a problem. This is CT that's high. But only when you sequence, you realize the diversity changes as well. So when you get a low infection, you get, and this is a pie chart, so every different color of a pie chart, of a wedge in this pie, is essentially a different virus genome. So when you looked at the diversity of the viruses in these environments here, we saw high diversity, low genetic load. Only when you find Varroa entering, you had a selection of a certain genotype, only 1 or 2. Previously, you would group them and say, oh, it's deformed wing virus causing the problem. In fact, only through sequencing we found it's not virus A, it's not virus B or C, it's deformed wing virus A. Okay, so there's a value in terms of not only understanding presence or absence or virus load with disease, it's actually which quasi-species is involved with a certain disease. And then you can design primers, and so we've just published a new primer set for you to go out and not again. So I'm talking to beekeepers. I usually say this to beekeepers. When the beekeepers need to go and look at the virus, they shouldn't just be looking at one virus, they should be looking at 3. And here in the US, the bees are predominantly infected by A. And this is probably the reason why the highest mortality of bees anywhere in the world is here in the US, and is less so anywhere else. Won't go into that. What I will say is a cursory look at PERS virus. I can start seeing, and colleagues within the VDL can start seeing quasi-species coming through the sequence data that's currently being generated. Here's just one example where you can look at one farm, and with one farm over time, you can see selection going from one quasi-species to another, to a third. And actually the answer lies not just looking at one gene, it's actually looking at the full genome. And in fact, if you had to sequence this farm using the open ring frame 5, which everybody seems to be using, looking at the variation here, you would say this is one virus. You would say it's the same virus. In reality, it's not. There's at least 3 different viruses here. It becomes even more complicated because these individual strands here are consensus viruses. If you look at the sequence that underpins a lot of these these consensus viruses, there's massive variation. So where you see these different colors and there's a consensus, this essentially means there's variation in sequence, which just makes up this consensus, which we compare to everything else. So while we try to oversimplify things and trying to make correlations between presence and absence and how these viruses are related, we do this by generalizing. And this is something that should not be done, because the devil is in the detail. And bioinformatically, we think now we can start finding out the devil, I guess. Maybe that's it. As again, I want to just thank you for your attention. I'm here because of a GREET and because of the University of Minnesota, and it's all funded through the Department of Agriculture. And maybe next year I'll give you more information about quasi-species and PERS.